Hey guys, this is the Bio 25 final exam review, and we're just gonna go ahead and start with some announcements first. Okay, so if you're in Bio 25, you're in the right place. This is the review session. Just wanted to point out a few things so that you guys are aware of it. Your final opens tomorrow, I believe, at noon, and then closes on Wednesday at midnight. So that gives you 24 hours to be able to take the final. Make sure you remember to take the final. There are people who forgot to take it before in the past. So just make sure you have the dates in your agenda planner, something like that. Another thing I wanted to mention was that Laura replaces your lowest exam grade with the final if you perform better on the final, which usually is a trend that she sees in students. So that's a good thing for to know. Another thing I want to say was the review session today doesn't quite cover all of the concepts that was going to appear on your final exam. So just make sure that you studied before you take the final. We can't cover everything because there's a lot of stuff from week 1 to up till now. But definitely use your notes, class resources, and you guys can refer to the TA weekly videos that we post on YouTube for explanations on concepts. And hopefully you guys were able to do some of the extra credit surveys I think Laura posted a week ago or a couple of days ago, because these count towards your overall grade. And another thing I want to mention was, in addition to this review, we also did like a regular style, like lecture type of final exam review session from last semester, which covers exactly the same material. So we have like a link to the videos, and just gonna click it so you guys can see. But essentially, it's pretty much delivering you the material we're gonna go through today, but just in lecture style. So if you prefer to watch those kind of videos, you'll see those here. So you click the link. Yeah, and I think that's all I have to say. Oh, and good luck on the final when you guys take the final. Make sure you get plenty of rest. Try to study beforehand, eat a good meal. And another thing I wanted to say is make sure that when you're studying for the final, try to, I know it's hard because it's like it's not in person, but I would say try to prepare for the final as if it was in person. Because if you prepare for it that way, then it's going to take you less time to work through your open response questions, because you'll have somewhat of an idea of what you're gonna say for the final. Do any of the other TAs want to mention anything?
Yeah, I think I just want to say that the final, I think I looked at or I saw an email that Laura sent out. The exam, I'm pretty sure opens tonight at like 12:01 a.m. or something like that. So you could start it as early as like midnight tonight if you wanted to. But yeah.
Oh, okay. That's good. Yeah. So if anybody wanted to start it at midnight, you can. Um, yeah. So maybe I just write that in so people don't get confused. But yeah, the exam opens today midnight. So yeah, so exam opens midnight up until Wednesday midnight. So yeah, in a couple hours then. Oh, and another thing I wanted to mention before I forget, I believe your final exam this time is a slightly different format. I think you guys are gonna have like part multiple choice and part open response. So it's gonna be different than your other exams, which were like all open response. So for those of you that like multiple choice, that's good news. But yeah, so that's all I have to say for the announcements. Next slide, let me see. Oh, and the next slide, I was going to show you guys something that Laura has for this class. So I don't know if you guys have seen the syllabus or peeped at the syllabus yet, but there is the final exam substitution policy. So essentially this means that if you perform better on your final than your lowest exam grade, then Laura will take your final and essentially count that count your final exam will like replace your low grade, but your final still counts. I don't know if that makes sense, but I just have an example here. So this student, we'll call it student A, they got an 80 on exam 1, a 78 on exam 2, then the final they got a 75. So based off of Laura's final exam substitution policy, this student wouldn't receive like a change in their grade. So this is the first example. And so compare that to student B. So this student got a 73 on the first exam, an 80 on the second exam, but on their final, they got an 85. So that would mean that this student's essentially their lowest exam score, which is their first exam, would be replaced by your final. But then your final also still counts as your final. Like it doesn't mean that one of your exam grades gets dropped. It just means that it is substituted for the lowest. So as you can see here, the grade that they got on their final counts for the grade for their first exam. So yeah, this is just explaining how the exam policy works and for this class. So does anyone have any questions on that so far? Does that make sense? Can someone give me a thumbs up if it makes sense? Thanks. Perfect. Thank you. Okay, so there's that. And yeah, these slides were just showing you guys like the content from each exam, but we're gonna cover most of these in the Kahoot game today. So this is just the exam 1 stuff. And then this is the exam 2 stuff. And then this is the new material. So I guess call that like exam 3 or like final stuff. Okay, so those are all the announcements I have. And now we're gonna start with the Kahoot game. I just have to get the link up. Let's just give me a sec. Does anyone have any questions so far? Okay, so just give me a sec. How do I do this?
I think if you log into your account, you could go to like the game that you made and then click play.
Okay. I want the questions in order, but how do I do show questions and answers on player's device? Yeah, okay. Okay, so I think it's just this. Great, so I don't know if like anyone here hasn't played Kahoot before, but basically all you have to do is put in the game code you see. Oh, first you have to go to kahoot.it. So if you have a cell phone or something like that, or an iPad, you go to kahoot.it, which is the link above here. And then you're going to put in, it'll ask you to put in a nickname. There you go. And then you'll be able to be part of the game. Yeah, and I think they have an app as well, which is nice. Yeah, let's see. There is minus TAs. There is what, 10 of you here? So just wait for people to jump in here. Okay, perfect. So we can start. Good luck. Make this full screen. Okay, so this is your first question. Okay, how do I do this? Does anyone want to explain why the answer is both tertiary and quaternary structures? Which most of you got correct. If anyone wants to put it in the chat or unmute themselves to explain why that's the answer. Anyone?
Well, disulfide bridges are like created between R groups in the tertiary structure, and those like carry over to the quaternary structure.
Yeah, that's correct. Yeah, so Haley basically said what I was gonna say. So in tertiary structure, the focus of the types of bonds you'll see are like between the R groups, and so these include bonds such as disulfide bridges. And I don't want to give away the rest of that because there's more questions about this, but disulfide bridges is included in one of the many bonds for tertiary and quaternary structure. So does that make sense to everyone? Can I get a thumbs up from you guys if it makes sense? Okay, perfect. So now we're gonna just go to the next question. What does show media mean? Okay, never mind. Next question. Oh, okay. Okay. So yeah, most of you got the answer correct. Does anyone want to explain why it's correct?
Isn't it because like hydrogen bonds are technically like really weak for the point of it needing, like DNA needs to denature, so like hydrogen bonds can't be that strong, otherwise the DNA wouldn't be able to come apart. And ionic bonds are like easily disrupted by changes in like uh temperature or their environment, because they're only slightly stronger than hydrogen bonds.
So I think you're correct about that. So I'm not too sure about like the DNA. If any of the other TAs want to talk about that, but I know what you were saying about ionic bonds is correct. They're um, definitely affected by environmental structures. But basically for this one, the covalent bonds essentially for this class are stronger than non-covalent bonds. If any of the other undergrad TAs want to add to this question, because I'm not sure how to explain for the hydrogen, if anyone wants to talk about that.
Yeah,
Yeah, I don't know much about the hydrogen in regards to DNA binding. I don't think you would need to know that.
yeah, I was just gonna say, I think in regards to hydrogen bonds, it's more like they were saying for DNA, since the hydrogen, like in DNA, you want to obviously like separate and come together as needed. So unlike covalent bonds, which are usually in between like backbones of DNAs and things like that, where you want to keep it really stable, the hydrogen bonds, you don't want them to be too strong. Otherwise, like it makes it hard to replicate DNA easily.
Yeah, so I think you're correct about the hydrogen bonds. Sorry, I just like for this class, I don't remember learning that, so I just wanted to confirm. But essentially the idea for this question is that your covalent bonds for this class are the strongest kind of bonds that exist for the purposes of this class, and covalent bonds are stronger than non-covalent. And the two non-covalent bonds in this question are H bonds and ionic bonds. Okay. Cool. Can I get a thumbs up if that made sense, or do people need me to re-explain? Okay. Perfect. Thumbs up. Makes sense. Okay, next question. Oh, okay. Great, next question. There are a lot of questions. Oh, everyone got that one right. Okay. So does anyone want to explain why that answer is correct? I mean, I could explain, but I just like you guys to explain so that we know you guys understand. You all got it right, so you must know why it's correct.
The amino acid, like each amino acid is like the monomer of the polypeptide, so they have to be connected in primary structure, and the reaction that's done is a dehydration reaction, which creates peptide bonds between the amino acids to create a protein.
Yeah, so that's correct. But yeah, for this is it to do with proteins level of structure, which I think was week 1 material, but essentially primary structure is basically your amino acids and like the way that they're connected together. So each of the amino acids are linked together by peptide bonds. And can anyone tell me whether peptide bonds are covalent or non-covalent? I gotta check the chat. Okay, covalent.
Yeah, covalent.
Okay, perfect. 3 of you said covalent. Awesome. All right, next question. Some of you are climbing up the leaderboard here. It's good. All right. Oh, let me see. Mixture of answers here, but the correct is the green. Anyone want to explain why it's hydrogen bonds? No? Any of the TAs want to explain why it's hydrogen bonds?
Would it be for the same reason as we were saying for DNA, so they're easier to come apart and denature and such?
Yeah, so essentially, these ones are kind of hard to explain because they're kind of like sort of definition, more like type questions. But for primary structure, it's a sequence of amino acids, and those amino acids are linked together by peptide bond. And then you have like enough amino acids, like thousands and thousands, like in a row, and those will form structures like beta sheets and alpha helices. Hopefully these names ring a bell. But if you after you have like a whole bunch of amino acids, like thousands and thousands, they will link together and form beta sheets and alpha helices. And the alpha helices, like if you think of DNA, it's kind of like a spiral staircase. And like the beta sheets are just pleated sheets. And those are held together by the backbone atoms, which are held together by hydrogen bonds. So can anyone give me a thumbs up if that makes sense? Thank you. Okay, next question. Unless people have questions, you can always stop to ask questions. Oh, leaderboard's changing. Okay, almost everyone got that question. So basically for this one, the R group of an amino acid is like a variable group, which means like it changes between each amino acid. And amino acids are composed of an amino group, carboxyl group, and they have like an R group, which is just like I think they call it this. It's also called like a side chain. And so like a amino acid, whatever the amino acid side chain or R group is, it's what gives it the characteristic of being able to classify it. So the R group could have like non-polar elements or polar elements. And so like the R group itself is what distinguishes one amino acid from the other. Can people give me a thumbs up that that makes sense? Thank you. Thanks. The thumbs up helps. Okay. Cool. So next question. Oh, everyone got that right. So a single polypeptide can contain multiple independently folded functional domains. Does anybody want to try to explain why that one is true?
I'm pretty sure that there can be different domains that fold independently of each other in a single polypeptide, and like each of those domains can have a different function, so it kind of makes sense that they would fold independently of one another. That's not really like a complete explanation, but I don't know.
No, that was a good explanation. So essentially, like a domain is the way to explain a domain. I'm just trying to think. So a domain is when you have like a polypeptide. Not a polypeptide, like I feel like it's just like a subsection of polypeptide that has its own function, and domains are typically like individually folded from the rest of the polypeptide. I feel like to explain this one, I need a picture. Let me see. I have something. Hang on just a sec. So hopefully everyone can see this, but domains is from like the week 1 material. Essentially, domains is just like an independently folded area of one polypeptide, one protein, and that area has a specific function. And so like domain you can see on the right side is just like on the right side, this part of the diagram is just one large protein, but there's 2 domains, so the green and the red. And so each of those domains is sort of independently folded from one another, and each domain has a different function. So like maybe one domain like phosphorylates is in charge of phosphorylating something, and like another domain puts ubiquitin groups on something. So they could just have different roles. And yeah, so that's domains, which is separate from subunits. And subunits are when you have like a protein, when you have like a large protein, kind of if you picture hemoglobin that has like several, 2 or more polypeptides, and 2 or more like separate polypeptides can make like a large protein, and each of those polypeptides within it we call a subunit. Did that explanation make sense? Or people lost? Okay. And if any of the other TAs wants to explain that, feel free, because I just want to make sure people understand.
I think you're good. I think that was a good explanation. Obviously, if anyone still doesn't really understand it, you can type it in the chat and then like I can explain it further while we go on with the Kahoot.
Cool. Yeah. And just one more thing. An analogy to think of when you think of domains and subunits, think of like one person. So let's say this person is me. I'm just standing there. I'm a person. I have like an organ system, and I have like individual parts, so arms and legs. And those individual parts have like, and myself, like I have different functions. So those would be like domains compared to like if I'm like one protein, compared to like if I went and I held hands with Tina and like a couple other friends, then like each person represents like a subunit, but within like one large protein. So I don't know if that makes sense, if it just complicates things. But like within each person, they could have like a domain that serves like a specific function. So if people want to give me a thumbs up that that made sense, or if they need me to explain it again. Okay, perfect. I'm just gonna keep going with the Kahoot then. Perfect. Cool. Oh, this next one is on signal sequence stuff. Okay, so most of you got this one right. Can anyone explain to me why the answer is false?
Because having an ER retention sequence means that the protein would then get held in the endoplasmic reticulum, and therefore it would fail to be secreted outside of the cell.
Sorry, can you say it one more time? Could you repeat it one more time just to make sure I heard?
Okay, I said like, isn't it because it with an ER retention sequence, it means that the protein would remain in the endoplasmic reticulum instead of being secreted outside the cell?
Yep, that's correct. So yeah, so essentially for a protein to be able to leave the cell, it would just need to have I think the ER retention sequence is not necessary. So like by default, when the protein is being trafficked, for it to leave the cell, it has to go through the ER and the endomembrane system. And so like by default, a protein setting is like if it doesn't have any other sequence, if it just has ER localization, it's going to travel through the endomembrane system until it like eventually gets secreted. So yeah, so that's why if you have any retention sequence anywhere, like in the ER or Golgi bodies or anywhere else, then it's not going to be able to exit the cell. So did that make sense for people? Okay, I see one thumb up. Okay, so next question. You guys are doing really good at this, by the way. So let me see, most of you guys got this right. So can anyone explain why it is false?
Cells will have multiple receptors on its surface, that way that it can respond to multiple different signals, ultimately making multiple different pathways in the cell functional at the same time.
That's correct. So cells can receive many signals like at the same time, like they're always continuously receiving signals. Like right now, your cells are receiving signals for survival, differentiation, dividing. It's just normal for cells to have signals all the time. And that's why it is hard to like study human cells outside in like a laboratory setting, because when you put cells like in a lab, like you're removing them from like all receiving all their signals at once. So it's hard to like keep them alive. So essentially cells receive many signals at many times, and there's no errors in signal transduction or anything. Like they can always receive many signals at the same time. That is correct. So does that make sense for everyone? Thank you. All right, next question. Okay, gonna click on the next one. Okay, so answer to this one is true. And it is because domains are highly conserved, meaning that between like different species, domains usually serve the same function. I can't remember if she went over this one in class, but I remember learning it when I took the course. But yeah, so just know that like domains can be like similar in function between like different species, that they're just meaning that they're like highly conserved. So does that make sense for people? Okay, cool. Thank you. Oh, so this one has more than one answer. Oh, okay. So it looks like most of you got this one. Does anyone want to explain why those two answers are correct? The bottom two?
I think based on what you said before about a domain, how a domain consists of like a fully folded polypeptide. I mean that is folded independently um, from the other polypeptides. So you also said, I mean, in a domain, and each polypeptide chain is a subunit in the domain, I think.
Yeah, so basically for subunits, subunits are involved in quaternary structure, and um, subunits, the way I think of them is like if you have like a mega protein made of multiple polypeptides. So if you think of something like hemoglobin, it just has like um, many many subunits attached to it, and they're like different polypeptide chains that make up the overall protein. So hold on, I think I have a diagram of that somewhere just to see that it like makes sense. So I don't know if all of you know about hemoglobin, but the way hemoglobin is made, or the way hemoglobin is designed, it has like the 4 subunits. I have a good image. The way hemoglobin is designed, like hemoglobin is a protein. So it's like a protein, and within the protein, there's 4 subunits. So you can see like the A chain and the B chain, and they're different colors. And 2 or more subunits is what we call quaternary structure. And so in a difference to like tertiary structure would be when you only have one polypeptide, whereas like this is quaternary structure because you have multiple polypeptides forming like a larger protein. So can people give me a thumbs up or unmute and say that it makes sense? Or if people still fuzzy on that? Okay, so it makes sense. Cool. Excuse me. Oh, everyone got this one. Okay, so someone can explain to me why this is right. Anyone want to try to explain? Even you can type in the chat. You don't even have to unmute.
Let me try. So activation energy is the energy required to carry out a reaction. So an enzyme does is the enzyme lowers this activation energy so that the reaction doesn't need or require enough. I mean, doesn't require a lot of energy to do the reaction.
Yes, so I believe you're correct on that. And so, yeah, so enzymes lower the activation energy, and activation energy is just the energy required for the reactants to be able to undergo the reaction. And so enzymes help facilitate the reaction to go smoothly by lowering the activation energy. And yeah, so enzymes only have the ability to change activation energy, and change is done by lowering it. So enzymes don't like increase energy in the reactants or products. They don't like lower stored energy. So anytime you just see enzymes, just think of like activation energy, because that's the only thing that they can directly affect. So does that make sense for people? Okay, cool. Okay, so does anyone want to explain why that answer is correct?
I'm not entirely sure, but I think it's because aren't chaperones like don't they like help the refolding of unfolded proteins? So essentially like a cell would essentially create chaperones or indicate the need for chaperones in order to deal with these unfolded proteins.
Yeah, so that's correct. So yeah, so the unfolded protein response, if you guys remember, is when you have kind of chaos happening. I can share this screen with to show this, but when you have sort of chaos happening in the cell, and this happens in the ER, by the way. And so when you have proteins that are misfolding or like not folding correctly, this is going on in the ER. And so what will happen is the cell tries to do things to be able to fix that. And so for this question here, one of the things this cell does is it decreases translation in the ER, because when you have proteins that are misfolded, you don't want to be creating more proteins, because that's just going to add to the problem. So the cell, what it does, will decrease translation to reduce the load on the ER. And then another thing it will do is something called increasing lipid synthesis, which means that the cell increases the volume of the ER so that it's able to accommodate for the misfolded proteins. So essentially, it's just expanding the volume of the ER to account for the increased protein load. And then just as I think Tati said, the chaperone proteins are your proteins that go around and help the misfolded proteins refold correctly in the correct orientation. And so you want to recruit more of those when there's the unfolded protein responses going on. So yeah, so that is why the square, the green option, is the correct answer. So hopefully that made sense. People want to give me thumbs up or stop me for questions. Okay. Okay. Most of you got this one right. Anybody want to take a stab at why this one is correct? Why, not why it's correct, sorry. As to why the option highlighted is correct.
I believe the more stable the bond is, like the higher, like the harder it is to break. So like covalent bonds are a lot more stable because they're stronger. So they're harder to break with heat than like non-covalent bonds. And non-covalent bonds are the weaker bonds out of covalent and non-covalent bonds. So the heat will damage them instead of like the covalent bonds.
Perfect. So that's the reason why. So essentially because the environmental factor of heat can have like extreme effects on non-covalent bonds, and non-covalent bonds, if you guys recall, they're much weaker than the covalent bonds. So that is why they get damaged. So that was a good, really good explanation for that. Okay. And if so, people want to give me thumbs up that that made sense. Thank you. And next question. And this one's kind of related to the last question. Okay, anyone want to try and explain why this one is true? Okay, so it's okay, I'll explain. So for this class, covalent bonds are our strongest bonds. And when we're looking at environmental factors that can affect proteins and things like that, covalent bonds are very strong. And so for this class, you just have to know that covalent bonds are strong enough to kind of like not be broken down by environmental factors, so the example being heat, unless it's given to, unless the protein is undergoing like a very highly extreme form of heat that's like very extreme. But for this class, you're just looking at it in the context of just like not the extreme forms of heat. So for the most part, like covalent bonds are strong enough to withstand heat. And so just heat in general will not break down covalent bonds. And so in the question I put thinking of an egg, because I don't know if you guys ever like eat eggs or cook eggs, but if you notice, like when you cook an egg, it just kind of crumples together. But the eggs, like because eggs are proteins, like the egg still stays connected with each other. And that's because like the covalent bonds are strong enough to withstand like the effect of heat, but your non-covalent bonds in the egg like are being heated up. So it causes the egg to sort of change shape and crumple up, and like that's how you get scrambled eggs pretty much. But the covalent bonds are still there. So you still have like your peptide bonds. And I think like if the egg, I'm not sure if eggs have disulfide, but like your covalent bonds, like peptide bonds, the amino acids are so strong. So the egg is able to, like, not all of your egg like breaks up in tiny pieces. That's why you get like large clumps of eggs. So can people give me thumbs up that that made sense? Or did the egg analogy help? Okay. Cool. Okay. So yeah, so just remember for this class that covalent bonds are strong and that they can withstand the effect environmental factors of heat. Okay, everyone got this one right, which is great. Anyone want to explain why that one is true?
I can try. So in the lecture she gave, I remember she gave an example of an ionic amino acid and she mixed it out with salt. And then I think the positive, um, sorry, the positive sodium bind with the negative part of the amino acid, and then the chloride, the negatively charged chloride bind with the positive side of that minority, hereby it causes the bond to be displaced.
I was not, I don't know if that's like I don't want like the details of it, but I think just for this question, I think it's just one of those ones where just knowing that pH and salt are able to have an effect on electrostatic interactions. And this is because our cells, like human cells, they operate at a certain pH and within the certain temperature range. And cells have the ability to control the salt around them. So just sort of tweaking up, increasing levels of salt, or even just fluctuating the pH just slightly can cause like non-covalent bonds, such as the ionic bonds, to be harmed in the process. So that's the reason why like the ionic and electrostatic bonds are affected by pH and salt changes, because the cell like has its own innate ability to regulate those. So deviating those from normal can have harmful effects. Did that make sense for people? Can people give me a thumbs up that it made sense? Oh, and someone added in the chat. Yep, ionic bonds are sensitive to environmental changes and will become disrupted by changes that change ionic ratio. Yeah, so it's just the fact that the cell is sensitive and that tweaking the pH that it normally operates at can cause problems. Cool. And you guys can always stop me if you have like more questions. Oh, okay. Most people got this one right. Anyone want to try explaining why it would be ER?
Even though it has both localization sequences, it essentially will get to the ER first, and therefore it'll probably stay there, because once it's in the ER, it's not really going to leave to then go to the mitochondria.
Okay. So yeah, so you're correct. And it's because like the proteins that end up in localized to the ER, while the protein's being translated, so while at the same time it's being translated, the protein's sort of being pulled through into the ER. And so because of that, once the protein's fully translated, it will already all be inside the ER before the mitochondrial sequence is read on the protein. So yeah, it just has to do with the mechanism of the way things are localized to the ER and the fact that the ER localization sequence is in the N-terminus, which means it's at the beginning of the protein. So that's the first thing that gets read by the signal recognition particle, which looks for that sequence and brings it into the ER. So yeah, so basically it's more to do with like just the mechanism of it and timing. So by the time like all the proteins being synthesized, it's already being pulled into the ER. And so by the time it's fully created, it's already inside the ER before like the second sequence is read. So does that make sense to people? Okay. One person told me it made sense, so I just want to make sure. Okay. 3 people. Okay, cool. Okay, almost everyone got this one right. Does anyone want to explain why it is the circle as the correct answer?
The NLS is needed to move the protein into the nucleus from the cytosol. If it needed to be moved out of the nucleus, then it needs another signal sequence, which is the export sequence. So because it doesn't have an export sequence, it won't be able to move in and out. It can just go in. So it'll probably just move from the cytosol to the nucleus.
That is correct. So basically, you just said it perfect. But if a protein, so when a protein is heading towards the nucleus, that's its destination. It needs to have NLS, which is nuclear localization sequence. But if your protein needs to ever come back out of the nucleus at any point, then it needs to have the NEX, which is the nuclear export sequence. Yeah, so that's correct. Okay, so this one is true, but does anyone want to explain why it's true?
Usually proteins that go into the ER that have like an ER localization sequence either have a retention sequence and stay there, or they're brought to a different protein in the endomembrane system, or they're exported. So normally they don't go back into the cytosol at all, even if they don't stay in the ER.
Yep, so that's correct. And that's everything you said was right. And I just want to point out another thing is that when a protein gets localized to the ER, the signal sequence is cleaved, so it's cut off, so that that protein does not is once it stays in ER, it's like stuck in that direction towards the endomembrane system instead of being able to come back out again into the cytosol. But yeah, usually proteins that enter the ER, they'll either stay in the ER, continue through the endomembrane system to enter some other membrane-bound organelle, or leave the cell. So does that make sense for people as to why it's true? Okay, it made sense to one person. Okay. Thank you. Great. So can anyone explain why that one is true?
If the protein wants to stay in the ER, it needs both a localization signal sequence and a retention signal sequence, because if it doesn't have a retention signal sequence, then it'll just get secreted from the cell. So it needs both to enter the ER and stay in the ER.
Yep, so that's right. So basically, if this protein has like ER localization and nothing else, then it's just going to go straight through the ER, then through the endomembrane system, like the Golgi bodies and that stuff, and then it's going to go on its way out into it's going to get secreted. So it just needs to be able for the protein, if it's like heading towards the ER, that's its final destination, then it needs the ER localization sequence to get there. And then it needs a sequence that says stop here, like stay here. Yeah, so hopefully that made sense to people. People want to give me thumbs up if that made sense. Perfect. Okay, cool. Great. So yeah, you can explain like the next few if you want.
Yeah, of course. Okay, that one's true. Does anyone want to explain why? If not, I can take a stab at it. I believe it's because if the protein is misfolded, then it cannot, it doesn't have that spot for important to bind to, and if that doesn't happen, then the cell has no way of knowing where to send the protein. Is that clear for everyone, or should I?
I'm sorry, can you say that again? I'm sorry.
Yeah, of course. So basically, like as you know, like proteins, their function is very much tied to their shape. So, say if a protein that is meant for the nucleus misfolds, then there's a chance that it doesn't have that pocket open for important to bind to, and without that signal, or without important being a signal, then the cell has no way of knowing where to send that protein.
Oh, and also the idea is important that for nuclear proteins, your signal sequence appears like on the surface of the protein, like on the exterior, compared to ER proteins, where the signal sequence is in the N terminus. So it's like embedded in I would say that's embedded in like the primary structure, like the sequence of amino acids towards the beginning of the protein for the ER proteins. So the ER and I think mitochondria also, they have their signal sequences like built into the primary structure, so the sequence of the amino acid, compared to nuclear proteins where you have the signal sequence is kind of in the tertiary structure, so the overall shape of the protein towards like the top or the surface. And so the signal sequence acts as like a binding site for important to bind to that nuclear protein and bring it to the nucleus. So that's why if a nuclear protein misfolds, it might not have the correct sequence on its surface for important to recognize and bring it to the nucleus. Did that make sense?
Yeah.
Thank you. Okay, cool. Oh, I forgot I put this slide here, but I guess it's like the next category is pathway stuff. So I put more time on the questions just to give people a second to look at the image and think.
Yep, there we go. So I can see there's a bit of a tie on this one, so I'll explain it. As you saw from the pathway, I don't know if we could.
I can put the image. Sorry, let me put the image of it because
Yeah.
hard to tell without the image what's going on here. Okay, here's your image.
Yeah, so as you can see, the BAD is kind of held in place by an apoptosis inhibitory protein, which when is phosphorylated, that protein is then free to go, and then you can see BAD is able to phosphorylate with another protein, and that will allow you to inhibit apoptosis. But if that sign never gets there, then the cell would just go its default pathway of cell death.
Did that explanation make sense for people?
Yeah.
Okay. Yeah, so it just like in the image, the bad protein's phosphorylated, and when it gets phosphorylated, that inactivates it, which allows the BCL2 to be free to inhibit cell death. But if that doesn't get phosphorylated, then it stays stuck to BCL2, and BCL2 cannot inhibit, BCL2 cannot prevent cell death, so cell death would happen. So that's why the answer to that is it would undergo cell death.
Okay, yep, the signaling would occur less often than normal. Does anyone want to explain that? Or I can, yeah, I can, excuse me. So pretty much the whole way that the GCPR pathway starts is through that dissociation of the G alpha subunit, as you can see there. Because then the beta and gamma split off into their effector downstream, and then the alpha splits off into his effector. So pretty much if it has a stronger affinity for the resting state, then it's going to be less likely that the alpha protein will be able to be free and interact with its downstream effectors. And also, like conversely, if the question had asked if there's less affinity, then the answer would be the opposite, where signaling would be more often than
So that makes sense for people? Okay. I have 2 people. Okay. 3. Perfect.
Yeah.
Oh, this question got it. Most of you. Anyone want to try to explain why it's false? Let me put up the pathway. Yeah, so GRAB2 is the adapter protein, which is the dark blue.
It doesn't catalyze any reaction. I guess the closest protein that would be an enzyme to it would be the RAS activating protein because it hydrolyzes the G or JK. That's not right either. But it's not an enzyme because it doesn't catalyze any reaction.
Yeah, so the GRAB2 adapter protein is not an enzyme because like for something to be an enzyme, it has to sort of catalyze some sort of reaction. So an example, I believe RAS would be considered to be an enzyme, right? Syed or Tina, am I right about that? Because it can hydrolyze GTP and a GDP on its own.
Yeah, because it has the enzymatic activity for hydrolyzing. Yeah.
Yeah, so basically for this class, like don't be fooled, not every protein is an enzyme. But just for something to be an enzyme, it has to be able to catalyze some sort of chemical reaction. So an example of an enzyme could be like a kinase, because kinases, even though they phosphorylate things, they sort of like catalyze like the reaction with like ATP to be able to get the phosphate. So there's like some, I don't know how to, like there's a reaction that goes on with like the like the ATP giving the kinase like a phosphate for it to add on. So I don't know if does that make sense to people?
Also, a hint is that an enzyme usually ends in ace. Biologists aren't really that creative, so they go with like these trends. So like helicase, kinase, ligase, things like that. Those are enzymes.
Yeah. Most of the time enzymes end in ACE, but not all the time.
Yeah.
Yeah. But does that make sense for people? Because a lot of you said it was an enzyme, so we just want to make sure that you guys can understand why it would not be. Yeah? Okay. Cool. I'm gonna find like an alternate explanation for that. And okay, most of you got that one correct. Can anyone tell me why?
Isn't it because the receptor is essentially a dimer, so that if the signal's in the form of a trimer, it wouldn't fit the receptor to bind to it?
That is correct. Yeah, so signal is always in the form of a dimer, usually not a trimer. So as you can see here, it's like two monomers, I think, make a dimer. And in most of these pathways, the signal is a dimer, not a trimer. So that is correct.
Yep, that's good. Does anyone want to explain why that's true?
I said true because different cells do have different downstream protein, so they'll respond differently to a signal.
Yeah, that's exactly right. It all depends on the pathways of the receptor cell. And a good example of this would be like a lot, like actually in the brain, like a lot of neurotransmitters will have an excitatory, the same neurotransmitter could have an excitatory effect on one neuron or one bunch of neurons, or it could also have an inhibitory function on another set of neurons.
Yep.
So
So,
it all depends on the,
yep. So that's why it's true. And also, I wanted to go back to the other question on the GRAB2 is an enzyme. Why it was false is because, like in pathways, you'll typically see kinases, and kinase is an example of an enzyme. And basically, for something to be an enzyme, it has to catalyze a reaction. So the reaction that kinases are able to perform is that they cleave off phosphate groups from ATP and attach it to like a substrate. So kinases will cleave a phosphate from ATP, which turns ATP into ADP, and it will attach it to like another protein. So that's what, like when a kinase phosphorylates something, like that's what's happening there. So hopefully that's like a better explanation for the other question. Sorry. So yeah, do you want to explain this one?
Yeah, sure. That is okay. Yep. Like I don't know if you guys remember from the diagram earlier,
Yeah, let me add the diagram.
but pretty much BCL2 is the one that's going to be attached to bad, and um, if it's what was the question again? If it was easier.
If bad is mutated so that it has a weaker affinity for BCL2.
Yeah. So if it has a weaker affinity for BCL2, then it's going to be more likely to dissociate. And if that happens, as you can see, when BCL2 is free, then it'll lead to an inhibition of apoptosis. Whereas conversely, if it was the opposite, if it was more tightly attached or bonded to bad, then the cell would go to cell apoptosis, the default.
Yeah, so if you inhibit apoptosis, which is cell death, then you get cell survival. So does that make sense for people? Okay,
Yeah.
cool.
Yep, that's false. It's a kind of a tie, so I'll just explain it. In the diagram, while it shows that it has a phosphate group, it's actually the addition of, or excuse me, it's actually the removal of phosphate from ATP that then activates the following kinase. So it's not always phosphorylation that activates or takes you from the next step. Sometimes it could be the opposite, actually.
Actually, hold on. Let me see. So the question is saying it's RAS activates MAPK3, which is, okay, so I'm just trying to show it in the diagram. So the question said RAS activates MAPK3, which is this one, by phosphorylation. And the answer would be false because in the diagram, there is no phosphate, which is the yellow groups attached to MAPK3. So that's like a hint. And then the second part of it is just because like for this pathway, like RAS doesn't phosphorylate things. And yeah, so RAS somehow like gets into close proximity with MAPK3, which is the MAP kinase, and activates it. It's not really known how it does that, but it does not phosphorylate MAPK3 because I do not believe that RAS is a kinase. I don't think so. If any of the TAs want to confirm that, but I don't think RAS is a kinase.
Yeah, I don't think it is.
Yeah, but just know that for this pathway, this is growth factor pathway or the RTK, RAS MAPK, that RAS does not do any phosphorylating here. It just will get into close proximity with the MAPK3 or bind to it and somehow cause it to become activated. But then after that, you have like the MAP phosphorylation cascade, which you can see with the phosphate groups. So can people give me thumbs up if that makes sense? Thanks.
There we go. Does anyone want to explain that?
Sorry, let me show the pathway. So this is the pathway to the JAK-STAT.
Yep. As you can see that stats 5 actually has two functions here. One is that it dimerizes with itself when it's phosphorylated. And then later on in the pathway with other transcription regulators, it'll start the transcription of the milk protein. So in that way, it is both a protein and a transcription factor because of its role in actually starting the transcription.
And yeah, transcription factors just essentially are proteins that can bind to DNA and I think just like assist with transcription. And transcription factors are able to enter the nucleus. And they're usually like within one of the last steps of like the pathways. So that's why STAT5 can be considered to be both a protein and transcription factor. And most transcription factors, I think, are proteins anyway. So hopefully that helps. Can people give us thumbs up if that makes sense? Made sense for 3 people. Okay. Another enzyme question. Oh, wow.
That's fine.
All right. So based on the question we did earlier, can anyone guess why RAN, which unfortunately I don't have an image of, but RAN is from the nuclear import, nuclear export, and RAN sort of behaves like RAS. So can anyone explain why RAN is the enzyme? Let me put a picture of RAS so you guys can see. Anyone want to guess why?
Let me try. I think is it because it catalyzed the reaction, like the conversion of a protein with GDP to GTP?
Yeah. Yep. So that's right. So RAN, so similar to RAS, like has the ability to like hydrolyze GTP to GDP, and so like it's able to hydrolyze GTP to GDP, but it has, because it's an enzyme, so it can catalyze that reaction, but it does so at a really slow rate. So in the nuclear import and export diagrams, like you'll see that it will enlist the help of the protein GAP to do that. So GAP sort of helps RAN to, it assists RAN in hydrolyzing GTP into GDP. And GAP doesn't directly hydrolyze GTP to GDP because RAN is an enzyme that does that. But GAP sort of makes that process go by faster. And so because RAN can intrinsically or on its own hydrolyze GTP into GDP, then that is what would make it an enzyme. And yeah, the other ones aren't enzymes because they don't catalyze things or catalyze chemical reactions. Does that make sense for people? And GPCR is not an enzyme. I know someone said that. And it's because GPCR just sort of binds to a ligand. But G alpha would be an enzyme, but not GPCR, because GPCR just in the pathway for, what is this? GPCR. The GPCR is just like a transmembrane protein, and the ligand binds there, and then causes stuff to happen. So hopefully that makes sense.
Oh, they got some people. Yeah, I feel like this one, It's always easy to mix up SH2 and SH3. For this one, you just gotta remember that for graph 2, it's going to be binding to the SH3 domains on the RAS.
Okay, let me see. So for this one, it was asking for what's the domain that connects the adapter protein or graph2 to the RAS activating. So this one's SH3. And SH3 domain, I think, is like just the I think it's just like the PXXP motif. So I think it's like a proline amino acid. The other TAs want to correct me on that. But this one would be SH3, but SH2 would actually be this one. So what connects the active RTK to the adaptive protein? Because SH2 domains is a site that has a phosphorylated tyrosine. And so this would be considered SH2 domain. And this one would be SH3. So anytime you see like a phosphorylated tyrosine or phosphate group connecting two things together, I guess most of the time it's an SH2 domain. I can show you guys another example of that. Probably be this is also SH2. What else? These two are SH2, phosphorylated tyrosines connecting the two together. But yeah, can anyone give me a thumbs up if that made sense or people lost?
So the SH2, is that in like the JAK-STAT pathway? I can't remember where like those domains are used.
Sorry, I should have put labels on these. But yeah, the SH2 domain, anytime that you see in a pathway, anytime in a pathway that you see a phosphorylated, like if you see the yellow P, like the phosphorylated tyrosine, like the SH2 domains are here and here. It's probably SH2 domain on the receptor connecting the STAT5. And then these two come together because of SH2 domains. So it's just anytime you see like an SH2 domain, I think like by definition is when you have like phosphorylated tyrosine area connecting two things. Did that make sense? I feel like I lost people.
Yeah, do people get that?
I feel like I lost people. I don't see a lot of people. Okay.
Makes sense.
Okay, thank you. Yeah, speaking of helps, because I can't tell. Can't see the chat and I can't tell. Okay.
Oh, good. Does anyone want to explain why that is?
I'm not 100% sure, but aren't there some cases where the signal crosses the membrane by itself and then it binds to a receptor that's like more like inside rather than on the outside?
Yeah. In like cases of if you think about like some hormones, like steroid-based hormones, they can just cross right through the cell membrane, and they don't need a receptor on the outside.
Yeah, so essentially, you can have like the extracellular receptor, which in this question is like a transmembrane receptor that's on the surface of a cell. But then you can also have receptors that are intracellular inside the cell. And yeah, and so if you have like a really large signal, I think it's signals that are large. I could be wrong. Signals that are large and hydrophilic bind to the extracellular receptor on the outside, and then small hydrophobic go in the nucleus, not in the nucleus, go inside the cell. Is that right to the TAs? Does that sound right? Or am I mixing it?
Yeah.
Yeah, that sounds right to me too.
Yeah, I remember she had a question on that too. At least when I took the class. On
Yeah, okay. So yeah, so if a signal is like really large and I think polar, then it stays on the outside of the cell and binds to extracellular. And then there are also intracellular receptors too. So there's not just one kind. I think that's why it would be false because it says always. So can people give me thumbs up that that made sense? Okay, yeah. Sorry, I was just checking the chat to make sure I didn't miss something because I can't see it.
Yep.
Oh, everyone got that one.
Does anyone want to explain why?
With the receptor always active, then it's always dissociating, which means that it's always producing a signal.
Yeah.
What specifically, just to make sure, what specifically is always dissociating? You remember?
Do you mind if I see the diagram again?
Of course, I'm gonna put the diagram because yeah, it's hard to remember. Okay, what specifically when you say, because you're right, but I just want to make sure. What specifically is dissociating?
So like the G alpha subunit is dissociating away from the like GBY subunit.
That's right. You're good. Okay, so basically the question is saying like if your GPCR receptor, which is the black part, if that's always active, always on, so instead of being in the resting state, it's just like it's always on. So like it's as if it's always bound to like a ligand or something, but just magically always on. So even if there's a ligand or not, it's just always active. Then these sequence of events will happen, and you'll have like once well it doesn't matter if there's ligand or not, but all the time like G alpha like will be bound to GTP, then it will dissociate and go into signaling, and like you have the active GPCR, and then eventually like this cycle will cycle back, and then you have like RGS binding to G alpha and helping it hydrolyze GTP into GDP. But yeah, and regardless, the receptor will always be active and always cause like signaling to happen. So does that make sense for people as to why that would be that answer? Thank you. Yeah, so if something's always constitutively active, then that means you just get like signal occurring in overdrive. Yeah. Okay.
Oh, good.
Oh, everyone got that.
Does anyone want to explain what the other kinds are? Downstream signal.
Or anyone could just explain like why they think the crosstalk is correct.
Well, crosstalk is basically like how the different pathways like interact with each other and like how they impact like the signal transduction pathways in general. Signal amplification is about like amplifying the signal, like making it using fewer like proteins or like proteins to make a bigger response. And then downstream signaling is just like the signal downstream of the pathway.
I mean, that's a pretty good response. But yeah, this is just like sort of like a definition based question, but I can see if I can get like an image but I can tell like you know what you're saying here. But signal crosstalk is just like when you have you can have two or more pathways that just kind of like interact with each other or communicate with each other. And so like you can cause like one pathway to become active and the other one inactive. I'm just trying to come up with like a good diagram to show you guys. But, sorry, I'm taking forever, but I'm just trying to get like a good diagram. Oh, okay. Here's something. So maybe you guys can see this kind of blurry, but you have like two different pathways and well, there's many pathways there. Hold on. So maybe this one's better, but you have like, you can see that like the yellow and the pink are like separate pathways, but like they have components in them that can communicate to either or components that are part of both pathways. So you see like this protein can be like activated by this one and this one, even though the yellow and the pink are separate pathways. So that's what we mean by like when we say like crosstalk, that one pathway can have influence over another to cause it to become active or inactivate the components that are inside it. So can people give me a thumbs up if that made sense? Okay, I see the thumbs. That makes sense. But yeah, you can see separate pathways, but they're able to talk to each other or quote unquote communicate. So that's what we call crosstalk. And I think there's a question on another term on here, so I won't ruin it. Just go to the next. Okay.
Oh, so it's a kind of a split.
Let me put up the diagram, sorry. So question says, when JAKs become phosphorylated, what happens next? And the answer was JAK proteins phosphorylate the prolactin receptor.
Yeah, I can see why people jump to the stats part, but that actually happens later in the pathway. First, the JAKs phosphorylate each other, and then they phosphorylate the receptor itself, and then you move on to the stats.
Yeah, so for this question here, it's just like the sequential events of the pathway itself. And so JAKs is different than the other pathways because the receptors are not RTKs. So I think that might have tripped people up a bit on that. But for the JAKs one, just the order of like the diagram, once the JAKs are phosphorylated, then they phosphorylate the receptor because the JAKs are just like added onto the receptor. It's not an RTK. And so once they phosphorylate the receptor, that makes a binding spot, SH2 domain for the Staph5s to bind to. And then, yeah, then the JAKs are able to phosphorylate the Staph5, and then like the rest of the pathway occurs. Did that make sense for people? Okay, 2 people of 10. 3 people. We'll take it. All right, stop us if you have questions too, because we want to make sure you guys like understand. Oh, okay.
That's good.
But yeah, anyone want to explain this one real quick?
If not, I can also. Yeah, it's like someone said earlier. Pretty much the difference between this and the crosstalk is that in single application activates a bunch of different downstream. I know the analogy Dr. Francis likes to use in class is like spreading rumors. Like one person talks to two people, and those two people talk to two people themselves. So then it kind of just exponentially grows from one starting point. So it's just another way for cells to save energy um, and be more efficient, pretty much.
Yeah, so amplifying something essentially makes it stronger, or like, so you just have like a one signaling molecule can cause like just the signaling to amplify. So you see one signaling molecule activates like one adenylyl cyclase, can't say that, which somehow activates cyclic AMP. Then like one cyclic AMP can bind to one protein kinase A, and one protein kinase A can activate like multiple other proteins. So you see how like in a pyramid effect, like the signal is just being amplified. Like one signal could have such a huge effect to cell response because you see cell response happening like in a large fashion. So can people give me thumbs up that that makes sense? Okay. Okay, pretty much everyone got this one. But it was what we were saying earlier, how in this JAK-STAT pathway, you have the phosphorylated tyrosines on the STAT5 come together, the SH2 domain sites bind with each other, and so that's what makes the STAT5 dimer. Does that make sense? Okay, cool. And okay. Syed, do you want to explain this one?
Yeah, sure.
So question says, if there's a mutation in AKT that causes it to only get phosphorylated at one SH2 site.
Yeah. So pretty much, like you can see from the pathway, it needs to be phosphorylated at both ends to be dissociated from P3. So if you only get one side, say if you only get the MTOR or you only get the PKD on one side, then it won't be enough for dissociation to happen. And if that can't happen, then it cannot phosphorylate bad, which would then lead to cell death.
Does that make sense for everyone? Why it would be cell death? One person.
Yeah.
Okay, thank you. So I was thinking, you guys, for time's sake, because we want to get to like the new material and stuff. Do people want to skip the pathway stuff and move on to microtubules and then start like going through the new material, or do people still want to keep going with the pathway questions? So we can just do like a thumbs up. So can people give me thumbs up if they would want to continue with pathway stuff and not move on to microtubules? Okay, nobody. And can people give me thumbs up if they want to just skip the pathway stuff, go to microtubules and the new material? Okay, that's pretty much everyone. Okay, so it must be a way for me to skip this. I mean, I think there's a way we can post this to you guys so you guys can like play this, the rest of the questions we don't get to. I'm just trying to see if there's anything. Because we did a lot of pathway stuff, so I want to make sure there's time for everything. Oh, okay.
Oh, good. I think most people got it, but for the people that said cuts and destabilizes, pretty much the way you want to think about it is that the way that Taxol is used is as a cancer treatment drug. So what it does is that by destabilizing the microtubules, they can't pull away as much during anaphase. And if that happens, then you pretty much can't have cell division, right? Because the chromosomes can't go to the separate poles. And if that can happen, then you're not going to get as much division in, say, a tumor, which is rapidly dividing. So that's pretty much the logic of why, or how to remember that Taxol is a stabilizing.
Oh, so this one has two answers. Syed or Tina, you want to explain this one?
Yeah, so pretty much, so GTP is right, what the, how would you say, the tubulin part that builds on the microtubules. So if that's gone, then you know, what is it? It's called a catastrophe, right? Where it starts to destabilize and break apart.
Yeah. Yeah, I think so. I think it's called catastrophe.
Yeah, yeah. So then you would have less material to actually keep building on the microtubule, and the length that you do have will then start to fall apart. Because also, as you know, that GTP will be turning into GDP, I believe. And when that happens, then it's not going to be bound as well.
Yeah, so the tubulin GTP, I think what you call it, tubulin GTP dimers, like they have strong affinity for each other. So like when you're assembling a microtubule, like you assemble the tubulin GTP dimers together, and they're strongly associated with each other. But eventually, like while the microtubule is being assembled, you have like GTP hydrolysis happening. So like eventually the microtubule will become like bound to tubulin GDP. And because you don't have enough GTP, like the GTP hydrolysis will catch up with the whole microtubule. And so like the microtubules like is poorly, the tubulin GDP will poorly associate with one another. And so that'll cause like the tubulin GDP dimers to fall off and kind of dissociate, which breaks the microtubule, which most of you got. So that's good. People give me thumbs up that that made sense. Thank you.
Oh, good. Most of you got it. Yep, some microtubules will grow and some will be shortened at different times.
And I
Because,
think this is just, oh, sorry, go ahead.
oh, no,
Sorry. It's just, I think it's because like microtubule growth is random. Like there's no structural
Yeah.
order, like not all of them grow and shrink. Not every single microtubule in a cell is growing at the exact same time, and not all of them are shrinking. I
Yep.
think that's why. So did that. Saeed, were you gonna add something?
Oh no, I was just gonna say that, like in any given like solution of it, some are gonna be coming together, some are gonna be coming apart. That's why in the cell you're gonna have that gamma ring as like a spot for it to build up, just to make it more efficient. But yeah.
Oh, okay. So this one is because microtubule growth is regulated by like GTP and GDP and not ATP. So that's why you wouldn't really, ATP doesn't really have an effect on microtubule growth. So that's why nothing would happen. Does that make sense for people? Can I get a thumbs up? Sorry, it was kind of a trick question.
There we go. Does anyone want to explain that? Or?
Yeah, it's just for this one, it's just colchicine is microtubule drug that, or is a drug that destabilizes microtubules. And then Taxol is a drug that stabilizes microtubules. I think this is one of your exam 2 questions, but that's just like the difference between the two. Oh, is this the same question?
No, this is Taxel. The other one was culture center.
Oh,
Which
okay.
you explained in the thing too.
Okay, so pretty much almost everyone got this one. But because not microtubules, because Taxol is microtubule stabilizing drug. If you add it to microtubules, you get more polymerized microtubules in the cell because Taxol encourages like microtubule growth. Does that make sense for people? Can I get thumbs up? Okay. Now just a couple more microtubule stuff, then we're gonna go to like new material.
Most people do that.
Yeah, this one is just like technique for measuring like polymerization of microtubules. It's just like a, I don't know, it's just like a definition one but you use spectrophotometry. And the other terms like don't make sense here. But yeah. So hopefully that made sense. Sorry, I'm kind of going through these because I want to make sure we get to the new material in the next Kahoot. Okay. Oh, everybody got this. And I think it's just because it's the only answer that makes sense here. But dynamic instability is like when you have the growth and shrinking. But yeah, the GTP, GDP have nothing to do with energy for tubulin. And it's not because that you tip the balance in favor of microtubule assembly, because microtubules can assemble or disassemble. There's nothing like that. And so yeah, I just think the one that makes sense is dynamic instability. If any of the other TAs want to add anything to that.
No, I think that's pretty much it.
Okay. Great. So last question for this section, then we're gonna do new stuff like cell cycle, material, and DNA stuff. Okay, so answer for this is kinesin and diamine because those are motor proteins that walk along microtubules and they can transport organelles. Does that make sense for people? Does everybody put that? So hopefully that makes sense. Okay, so this Kahoot's like is probably I'd say like 60% of what's on the exam. So we're gonna just switch to the second Kahoot, which has questions on cell cycle and like new material. So let me see. So Tina, if you want to put the link for this, for the new Kahoot.
Yeah, sounds good. Do you want me to just share my screen? I have it pulled up already.
Yeah, we could do that. But congrats to our top 3 winners and runners up. So I'm gonna.
Cool. So there's also a Sorry. There's also questions for the second half of this Kahoot, which will be like basic cell cycle material, and then DNA replication errors, like proofreading and all that jazz, DNA technology and such. And so I know we're hitting the 2-hour mark, but if you guys need to go, obviously feel free to go. We're going to move through this as fast as we can, but obviously feel free to drop it in the chat if you have questions, or stop me at any point, or ask the TAs questions, things like that. I can't tell if that's everyone. I think so.
Yes, it's everyone minus the
Okay.
TAs too. So
All right, sounds good. So first question, which of the following statements correctly describes P53? All right, exactly. So P53 activation of P53 leads to the inhibition of cyclin CDK complex. So P53 has a lot to do with, obviously, because if you stop cyclin CDK, then the cell cycle cannot continue on. And that is a big factor of P53. It plays a big role in like cell apoptosis and cell death. So any questions about P53? I think there's another question later on as well. All right. Okay, so the amount of nuclear DNA in the cell doubles during which phase? S phase, exactly, because that's the phase where replication occurs. So you go from the normal amount of however much DNA chromosomes you have, and then you replicate, so then it doubles during S phase. During which phase of the cell cycle do the sister chromatids separate from each other? So this was kind of a, I guess, trick question. I didn't make these questions, but Michelle, if you wanted to explain this, but I'm guessing it's because like the cell cycle M phase is part of the cell cycle. Anaphase is like a phase within the M phase. So I'm guessing that's where you were going with it.
I believe so. I think. Actually, I don't know. I might have made a. Let me just double check that because I might have made a mistake.
Yeah, but anaphase wouldn't be wrong. I guess it's just about the wording. So the M phase is part of the cell cycle, but anaphase is a phase within the M phase.
Yeah, so sorry to mess people up, but I think like I made a typo with this, but like I was going for M phase. Not that anaphase would be wrong. But I think like for this either or, but like just so you know, like Laura or Dr. Francis wouldn't confuse you like this. I just like made an error with creating the question. So yeah, the way I made the question, like the answer was supposed to be M phase, and like all the other ones were like not supposed to be the answer. But I think like in this case, either or, and metaphase or anaphase. Sorry guys.
No worries. If you guys have, if anyone has like questions about this, like feel free to ask about it as well.
My bad. Some people might have lost their score.
Oh no. It's okay. You can come back. Okay, during which phase of the cell cycle do pre-RCs form? Perfect, everyone got it. So the G1 phase, yep, the pre-RCs form in the G1 phase, and then that's because the pre-RC is needed in order to recruit everything to form the pre-IC before it can continue on into the next phase for replication. Cells that will never divide again are likely to be arrested in this phase. Yep, so the G0 phase. G0 phase is kind of like that phase that's always like apart from the entire cycle because in this phase, the cells are kind of just chilling, you know, like they're not really doing anything. They're not active, they're not being active per se or preparing or growing anything like that as they would be in G1. So they likely would never divide again. And most of our cells are in this stage, so. Chromosome condensation occurs at the beginning of this phase. All right, yeah, so M phase. So S phase, remember that's where you're duplicating all of your DNA, so you're not condensing it yet. M phase, the beginning of M phase would be prophase, so that's when chromosome condensation occurs. The nuclear envelope breaks into fragments during this phase. Yep, so M phase again, it would be beginning of M phase, so prophase, which is when the nuclear envelope breaks down for all the chromosome to come out. Telophase is the end of M phase where you start to have like the division, the actual division of the cells, and then anaphase would be like where the chromosomes are going apart away from each other. Again, I'm going sort of fast, but obviously feel free to stop me at any point. Exactly. So to be A, non-degradable cohesin and securin. So cohesin is what keeps the chromatids together, sister chromatids together. So obviously if you cannot degrade those cohesins, then the sister chromatids will never separate. So it'll be stuck at metaphase. And then securin, securin is what stays bound to separase. And so if you can't take away securin, separase will never be able to go break apart the cohesin. And then again, the metaphase point will stay because the sister chromatids can't separate. All right, so yep, everyone got this. CDK activity is at the lowest at the end of G1. So CDK, again, I know that this was a question before, but CDK is always there. It's just at different points. And then cyclin activity is what causes CDK activity to either increase or decrease. So just a quick question. I don't know if Michelle or Syed, if you guys can see, because I can't see people, but would people like to kind of like brush over, because I can skip over the cell cycle stuff and you guys can come back in to review it yourself. I can like upload the doc that I have with all the questions too, and then we can go straight to like week 11 and beyond, like past exam 2 things.
So thumbs up if people want new material, the new stuff, talking about DNA replication, what's the other
Okay.
stuff? Like recombinant DNA technology, all that stuff.
Yeah.
Okay, and then thumbs up if people want to stay on cell cycle and stay going through like how we are right now. Okay, looks like new material wins. So
All right, sounds good. We'll just move through and then I'll post things, like post all the questions later
Yep.
on in the night so you guys can go through them if you want.
You guys will be able to access this stuff, so don't worry. We're just skipping for time's sake to make sure we cover most things for people. There's just a lot of cell cycle, and I want to make sure we get most of the stuff equally. Yeah, so now we're just going to new stuff. People can put in the chat if they have questions, but
I don't know. I think there's like a couple more questions to skip through.
yeah, sorry. I mean, it's good news for you guys. You guys have a lot to practice from. Tina, you can pause the screen as well so that people like don't see that.
Oh, really? I can do that.
You can pause the screen somehow. So like
Um,
your answer until you're ready to go to like the next screen.
oh, okay. Wow, the more I learn. Okay, cool.
Yeah, so at least people can like practice.
All right, made it.
Oh, okay.
All right.
Solid.
So true or false, the 2' carbon in DNA is attached to a hydroxyl group or an OH group? Exactly, it's false. So the 2' carbon in RNA is attached to a hydroxyl group, which is why RNA is more reactive as like a single strand. But the 2' carbon in DNA is attached to a hydrogen, and only the 3' carbon is attached to an OH group, and that's what's like the bonds between the DNA backbone to form. Yep, everyone got this right. So you guys are good with the models. It's basically you'll have one original and then one newly synthesized strand stay together after synthesis. This is the conservative model where the original stay together and the new forms stay together. And then this is the dispersive model where you kind of have like chunks of the new and the old. All right, what is the bond between nucleotides called? Yep, so it's the phosphodiester bond. Phosphodiester bond is what the bond between the DNA backbone is. Peptide bond is between amino acids backbone, between like the amino group and the carboxyl group. So just be careful of that distinction. And then hydrogen bonds are between the nucleotide bases instead of the nucleotide itself. In DNA replication, DNA polymerase reads in the 5' to 3' direction, so it builds in the 3' to 5' direction. Is that true or false? Yep, so it's fall. It's actually the opposite. It reads in the 3' to 5' direction, so that it builds to the sorry, it builds in the 5' to 3' direction because it needs that open OH group on the 3' end in order to add the next nucleotide. So you always want to have that 3' OH open. DNA polymerase can't start complementary base pairing from nothing when it gets what gets recruited to the replosome site prior to building the DNA strand. Exactly. So it is a primase, or otherwise known as an RNA polymerase, that gets recruited in order to add on like a roughly 10 nucleotide primer made up of RNA for the DNA polymerase to build off of. It's not helicase. Helicase is what separates the two strands. How many replication forks does an origin of replication have? Exactly, two. So you have one on each end. If you picture like a bubble, there's like one replication fork on the left and one on the right. What are Okazaki fragments? Yep, so they are small pieces of DNA that are behind the leading strand, moving in opposite directions of the fork movement. So that just means if the fork movement is going towards the right, then the Okazaki fragments are moving in the opposite direction towards the left, and they're always behind the leading strand. So they are not moving in the same direction as the replication fork. Oh, so we're split 50-50. So the answer would be true. You would need, so the leading strand needs that only one primer, but the lagging strand needs a primer for every time it makes a new lagging strand or a new Okazaki fragment. So you definitely need to have the primase constantly there, part of the replosome, so that it can add a primer in at any point when it needs a new lagging strand. So for every lagging strand, you need a primer. So you can't just have like put one primer down and then the primase goes away. You would have to have the primase throughout the whole application process so that the primers can be added on for each Okazaki fragment. Does that make sense to people about why it's why you would need the primase there?
Yeah,
Alright,
I
so
see thumbs up. It makes sense.
cool. Okay, yeah, so it is this one. I think these two are similar, except this option has all of them in completion. So you need the helicase, that's what separates the strands. You definitely need the DNA polymerase because that is what synthesizes the new strand itself. you need a sliding clamp and the loader, because the loader is what brings the sliding clamp on, and then the sliding clamp is what allows DNA polymerase to be more stable on the strand so that it doesn't fall off as easily. You need the primase because in the previous question, the primase is what allows you to add those RNA primers in order for DNA polymerase to build off of. And then DNA SSBP is the single strand binding protein, because once the helicase separates the two strands, DNA as a single strand is very fragile and it would break on its own. But with the single strand binding protein, it allows the single strand to be more stable so that it stays separated and that replication can occur. How is the end replication problem solved? Exactly, yeah. So GCAP and poly-A tail are part of RNA processing, not in the end replication problem, which is that it has to do with like the NICs, and basically once you end replication, you want to add the telomeres, and then that's added by the telomerases, so that every time you replicate, some of the DNA on the chromatids can go away, but that's fine because the telomeres aren't the telomeres are not coding genes, so it's okay if some of those goes away. Whereas if you did not, because you would be chewing off the ends of your DNA, and then those DNAs are useful with coding as they code for genes. Okay, so this was kind of a trick question, but the domain, it is not wrong, but it is both the domain and the subunit, depending on the type of DNA polymerase. It could be a domain or it could be a subunit, but essentially the idea is that when you're proofreading, anytime that the DNA has a wrong base pair, it just kind of moves it from the polymerase active site into the exonuclease active site, choose back on that wrong pair, choose back on that wrong base, and then moves it back into the normal active site. So it usually is a domain, but it could be a domain or a subunit. True or false, mismatch repair occurs during DNA replication. That is true. Occurs post DNA replication. That is true because it searches for and fixes the base pair mismatches. So it would have to occur after DNA replication because it needs to differentiate between the new and the old. And obviously it wouldn't be able to differentiate if it does its job while DNA replication is happening. Okay, how do prokaryotes identify new strands from the parent strands in MMR? All All right, so newly synthesized strands are hemimethylated and old parent strands are fully methylated. Exactly, so everyone technically got it all right, but basically hemimethylated means that only some of it is methylated, not all of it, because there hasn't been enough time to go and fully methylated yet. So the prokaryote can just identify which one is not fully methylated, and then they'll identify that as the new strand to perform MMR. Okay, can MMR be performed in a eukaryotic cell if all the nicks in the newly synthesized strand are sealed? So the answer is no, because if all that is how eukaryotic cells differentiate new from old, the new strands are not fully sealed yet by all of the nicks. The ligase has not had a chance to go and seal everything yet. And so if all the nicks are already sealed, then it can't differentiate new from old, and then MMR cannot be performed. Okay, which type of DNA damage and repair mechanism involves DNA glycolysis? I cannot pronounce that.
Glycosylates.
Thank you. Exactly. Yep. So only one person got it right, but that's okay. So what DNA glycosylase does is that it creates an abasic site. And so for deamination, which is when your cytosine turns into uracil, the uracil is still there, of course. And so DNA glycosylase has to go in and cleave that uracil off to make it an abasic site in order for base excision repair to occur. Base excision repair starts off needing that, what's in the chat? Oh, that's okay. Guessing is okay too. But yeah, so base excision repair, or BER, needs to have an abasic site, which means that there's no nucleotide base there. So there's no A, T, G, or C, or U. And so depurination, you're already starting off with a basic site. So the purine is already not there. That's why BER can work, and it doesn't need the glycosylase to take that, that base away. Deamination, the cytosine was turned into a uracil, so that uracil base is still there. And in order for BER to work, glycosylase has to go in to take that base away. And then in pyrimidine dimers, it's an nucleotide excision repair. So it kind of takes like an entire chunk of DNA away rather than just one base. So it wouldn't be that either, because it doesn't need to make it abasic. And then depurination and deamination, again, it would not be this, it would be just deamination, because depurination does not need to create an abasic site if it already has an abasic site. Does that make sense? Or if people need me to like re-explain it slower?
It makes sense.
All right, sounds good. Cool. We're almost there, guys. Okay. In NER, you notice that a gene has nicks, but the damaged DNA is still present. Which protein was mutated to have caused this problem? Okay, so we're definitely split a lot here. So this problem would require you to understand which, like what everything does. So it would be a helicase, because if the gene has nicks, that means that if the gene has nicks, that means that endonuclease already went in to nick the DNA. So it would not be endonuclease because that's working. DNA out, and the protein that does that is helicase. So helicase is what removes the section of damaged DNA after the endonuclease has already cleaved on either side of it. DNA ligase would be at the very end, where after the new proper DNA has already been like base paired in, and then DNA ligase would go in to seal everything. And then sliding clamp, no one chose it, but sliding clamp is basically what recruits the DNA polymerase. So the answer is helicase because you nicked it, so the endonuclease is working, but the damaged DNA is still there. So that means helicase must have not been able to go in and take that damaged DNA out. All right, similar question. In nucleotide excision repair, a gene with a lesion has completely missing section of DNA. Which protein mutated in order to cause this problem? I think there are two answers to this, so multi-answer. Okay, so most people got it right, I believe. But basically, you would know that helicase is working now because unlike the other one, the missing section of DNA that was, or sorry, the wrong part of DNA is actually gone. So that means helicase did its job in taking that damaged DNA out. However, that chunk is still there and missing. And so that means DNA polymerase must have not been able to go there and create the new proper DNA bases. And if DNA polymerase was not there, sliding clamp also must have not been able to be there because that's what recruits and helps the DNA polymerase stay stable. So DNA polymerase and sliding clamp are kind of like a duo package deal where they stay together. DNA ligase would only be you would think that DNA ligase is mutated if it said something like, oh, the proper DNA has already been replaced and everything is fine, but there are still nicks. And so that's when DNA ligase would be mutated because it was not able to go in and seal everything in the end. Does that make sense? Do people have like a better understanding now of like what these proteins do? All right, I see thumbs up. Cool, perfect. Okay, unpaired lesions such as pyrimidine dimers from non-functional nucleotide excision repairs could lead to. So if the pyrimidine dimers or lesions are not repaired, what outcome could occur? Yep, so the answer is double-stranded breaks. One of the, I think this was just something that Professor Francis talked about like in her class, but with pyrimidine dimers, since they're so bulky and they're so chunky, if you don't fix them, such as if non-nucleotide excision repair, which is how you would fix pyrimidine dimers, if that's non-functional or if it's mutated or it's just not working and these lesions are still in the DNA, then it could lead to double-stranded breaks, which is not good. The DNA wouldn't exactly clump together because if the double-stranded breaks are occurring, your DNA would just kind of your DNA would end up dispersing, which obviously is not good because you don't want your DNA to be breaking, but it would not be clumping together. It would be double-stranded breaks because the pyrimidine dimers, these are like big, bulky lesions. And so it's not good for like the steric chemistry of DNA, which you don't need to know that deep. But yeah, just know that if these are not repaired, then double-stranded breaks could occur. All right, true or false? After exposure to UV radiation, a human has double-stranded breaks. Cells would most likely form HDR over NHEJ because HDR has less errors. Oh, sorry, it should be a human. Yeah, a human has double-stranded breaks. So think about that part. It's a human. Yep, so it is false. Humans prefer NHEJ, non-homologous end joining over homologous directed repair because it's easier, faster, and not as detrimental. Basically, humans have a lot more non-coding gene areas. So even though it has exonucleases where it would like chew back on a lot of genes, it wouldn't be that bad because it's non-coding. So it wouldn't really affect anything to the DNA. Also, humans don't have a lot of cells that are currently dividing, so there's no repair template available for HDR to occur. So HDR needs a repair template, but humans usually don't have any. Okay, BER and NER involve endonucleases rather than exonucleases, true or false? So it is true. Only endonucleases are used when they cut the DNA strand. So base excision repair or nucleotide excision repair, it just cuts, endonucleases cut in the backbone. Exonucleases are not used because they're not chewing at any ends. So exonucleases are at the ends, whereas endonucleases are inside or like in between the DNA. A vector containing foreign DNA is Yep, perfect. Everyone got that. Recombinant DNA. These are all just different types of DNA that you don't need to know. But recombinant DNA, once you put something foreign into another DNA, now you've recombined DNA. Okay, restriction enzymes are. So this is also multi-answer. There's 2 answers. Perfect. Everyone got that. So they are sequence specific. They cut at specific restriction sites. And then they are also known as restriction endonucleases because they cut, the restriction sites are always within or inside the DNA. It's not on the exterior. It's not chewing back on anything. Perfect. All right. Last few questions. You take an entire desired eukaryotic gene to be cloned in an E. coli vector. Will protein be expressed? So you take the entire eukaryotic gene into an E. coli vector. Yep, no, it would not occur because if you put the entire gene, that would mean you're including the promoter of a eukaryote. And E. coli's do not have the proper transcription factors or anything like that to process the eukaryotic gene with a eukaryotic promoter. So E. coli's are very E. coli specific. E. coli would need E. coli specific promoters. Yep, so E. coli, you want to put in a gene that has 10 introns. Can you just take that gene from any genome or do you need to isolate the mature mRNA? Also, sorry for the grammar. Kahoot only lets you put a certain amount of characters. Yep, so you have to isolate mature mRNA because E. coli does not have RNA processing, as we know. So in order to have, if you put in just the normal genome, it's going to have all the introns, which we don't want because introns are usually spliced out. But since E. coli can't do that, then we have to isolate mature mRNA with all the introns already spliced out. And then we can reverse transcriptase that into cDNA and then use the cDNA as the template for PCR. So similar question, reverse transcriptase uses blank as a template to produce blank. All right, most people got that. So yep, it uses mRNA to produce cDNA. So it is not, normally it's DNA to RNA, but it's reverse transcription. So you're going from mRNA to cDNA. So PCR uses thermal cycling with primers. Temperature has to be low for primers to anneal to the cDNA. It doesn't matter how low. So can you go as low as you want? Yes, it does matter, exactly. So if you go too low, then the primer will become less sequence specific, and then it'll bind to a region that might not be fully complementary to the primer. So obviously, if the primer is not in the right spot, then polymerase is going to start transcribing in the wrong area, and then you'll have DNA sequence. So too low is too low. What causes chain termination? Exactly. Yay, everyone got it. So dideoxynucleases, nucleotides. So normally you have regular deoxynucleotides. That's what normal nucleotides are called. But dideoxynucleotides means that both the 3' and the 2' don't have an OH group. And again, if you don't have an OH group, then there's nothing for the next nucleotide to be able to bind to. You have to have at least that 3' OH group available. In gel electrophoresis, DNA travels towards the blank end and the shorter or longer strands move faster. So is it positive, negative, or shorter or longer? So DNA is negative, so it's going to move towards the positive end because negative and positive are attracted towards each other. And then shorter strands move faster, whereas longer strands move slower. So you would see that shorter strands are more towards the other ends as they're moving faster, whereas the longer strands are further in the front. So if you put in 2 different cDNAs into 2 unique restriction sites, how many strands would you see in like a gel if recombination worked? Okay, so it would actually be 3. So if you picture like a circle, you put in 2 different cDNAs. So you would see 3 nicks. So you would have 3 different nicks. And then I don't know if, just really quick, because it's hard to picture, I guess. But if this is your circle, and then you have one section, and then 2 sections. So then you have 1 nick, 2 nicks, 3 nicks. So you'd have 1 strand, 2 strands, 3 strands. So these 2 strands would be the 2 new ones that you put in. That's why you would have 3 different sections or 3 different strands. And then if you have 3 different strands, that would show you that your recombination worked. Does that make sense? Oops. Okay, perfect. All right, last question. We made it. True or false? When performing Sanger sequencing, you want as much dideoxynucleotides as possible so that chain termination can occur. Exactly. Yay, we end up on a good note. So exactly, too much dideoxynucleotides would cause too much chain termination. So the whole point of sequencing is that you want to see, you want to have random chain termination, but not so often that you can never tell what the sequence even is. So you want to have kind of like that perfect amount. Too little would cause no chain termination to occur, but too much would cause too much chain termination. So right in the middle, in order to see what your sequence is, so that you can like pair up all the numbers together. But yay, thanks for everyone who stuck around this long. Again, I will obviously, or all the TAs, we will put in like send out an email with all of our information if anyone has questions. Otherwise, good luck on your exam, guys.
Best of luck, everyone. Thanks for coming. We'll put the videos all gonna up on YouTube soon, so you guys can like go through the questions we didn't go through, pause and check everything. But thank you.